Good morning and afternoon, ladies and gentlemen, and thank you for joining us. I am Felicia Hawthorne, your moderator for today's webinar. I am pleased to introduce Heather Wetzel, who will lead the webinar titled Augmented Exome Sequencing Provides Clinical Grade Coverage and Sensitivity, which discusses our recent publication in Genome Medicine. Heather is a genetic counselor and member of our field applications team who brings years of clinical experience from prestigious institutions such as UCSF, Stanford, and Northwestern. Please join me in welcoming Heather Wetzel.
Thanks so much, Felicia, for the introduction, and thanks to everybody for the time that you're spending today with us. As Felicia said, the primary purpose of today's presentation is to review the recent publication in Genome Web in Genome Medicine entitled Achieving High Sensitivity for Clinical Applications Using Augmented Exome Sequencing. So with that, we'll get started. So as you can see from the outline, I'd like to first begin by briefly explaining Personalis and our mission, as well as a small overview of our various applications. Then we'll go through some of the highlights of our recent publication and finish by applying the findings of the publication to the clinical world by showing a couple of case examples that really highlight our performance. Finally, we'll save some time for Q&A and discussion. So for those of you who may not be familiar with Personalis, we're a private company born in late 2011 and founded by John West, our CEO, and 4 professors, Mike Snyder, Russ Altman, Ewan Ashley, and Atul Butte, who's now at UCSF. We've grown quite a bit since that time. Most of our employees are housed in Menlo Park in Silicon Valley, which is what you see here. Although we do have a number of field-based employees anywhere from California to the UK to Australia. We have a CLIA CAP lab, and primarily we focus on cancer and Mendelian syndromes. So as we'll discuss in more detail, our entire library of services is really built on the ACE platform. And ACE stands for, as you see here, accuracy and content enhanced. And this platform, really every step of it, is the basis for everything we do at Personalis. We've got products for researchers doing large and small scale sequencing studies in complex disease and Mendelian genetics. And we're rapidly advancing our presence in cancer research and clinical trials for tumor profiling. And last but certainly not least, as we'll talk about mainly today, we perform clinical grade testing via exome sequencing, utilizing our ACE clinical exome, as well as our newer partnership with Athena performing the Neurome, which is a whole exome geared towards neurological phenotypes. So what you see here are the steps involved in next generation sequencing. When we developed the ACE platform, our focus wasn't just to accept that there are problems with next gen sequencing, but really to improve on each step of the process with the goal of improving accuracy throughout, starting with sequencing all the way through to interpretation. And so this is really the basis of our company. And our recent publication in Genome Medicine really focuses on the sequencing part of this process. So what we'll do next is review the background and findings from our recently published article in Genome Medicine. So this is just a snapshot of the first page of the publication, but a couple of points to note here is that our authors are all primarily employees of Personalis. Our co-founders are included in the publication as well. A couple of other names that I want to point out to you, though, are Justin Zook and Mark Sallet, and these are employees of the National Institute of Standards and Technology, or NIST. And we collaborated with these two for this publication, and we've also collaborated with them on a number of projects at Personalis as well. So just to go over a little bit of background about exome sequencing. As I'm sure most of you know, exome sequencing is really utilized more and more often in the clinic when there's an evaluation of a suspected genetic disease in a patient or in a family. It's increasingly resulting in diagnoses in the clinic, but unfortunately there's huge variation in the overall coverage and sensitivity of various whole exome sequencing analyses. And that's really evident, particularly over medically relevant regions of the genome that really aren't well understood. There's a number of assays out there that continue to provide coverage that's really inadequate for clinical grade testing. They may, for example, cover 90% of coding content on average, but they might leave wide gaps in really important areas. And this could be a huge mistake in the clinical setting where accuracy really impacts medical management in a patient or in a family, as well as testing decisions for that patient or other family members. So our purpose in this particular study was really to examine just that, to look at coverage and accuracy that have been obtained with these currently available exome capture technologies, as well as looking at whole exome sequencing, which is also considered for clinical applications. We really wanted to evaluate how well these assays reach a clinical standard of coverage in order to interpret important genes of the exome, but also medically important genomic regions, as I've already mentioned. And of course, finally, we want to use this information to be able to provide you, the clinician, with information to help in evaluating and making decisions about genome scale testing in the clinic for your patients and families. So to do this, we use sequence data that were all obtained from the same reference sample, NA12878. We use predefined list of medically relevant protein coding and non-coding sequences. We captured coverage breadth and depth among 4 commercial exome sequencing platforms. And, as we'll talk about, these are platforms that are commonly used in molecular genetic testing labs. And we also looked at whole genome sequencing. And of course, we also evaluated the performance of our own augmented approach, which we've called ACE. So what you see listed here are the platforms for which we obtained sequencing data and the acronyms that we use throughout this talk, but also in the publication itself. So I'll kind of walk you through what each of these are. Of course, at the top is ACE, which we've already mentioned, is our Personalis accuracy and content enhanced clinical exome. SSCR is Agilent Sure Select Clinical Research Exome. SS is Agilent Sure Select version 5 with UTR, which is untranslated regions. NX is Illumina's Nextera. NG is Nimblegen's SpeakCap version 3. And of course, WGS is Illumina's whole genome sequencing at 31x average coverage. I should point out here that these 2 through 5 here, these are platforms that are the sort of standard platforms used in other clinical service laboratories as their exome sequencing capture kit. So a short background on ACE. We've mentioned ACE a couple of times here and there. So just to sort of walk you through a little bit more on what we mean by our augmented exome. As I mentioned earlier, ACE stands for Accuracy and Content Enhanced. And its purpose is to really address coverage and accuracy issues that are found in conventional whole exome sequencing tests, as well as whole genome sequencing. With ACE, we perform standard whole exome sequencing, as well as targeted enhanced coverage of medically relevant genes. And so for ACE, we're looking at approximately 8,000 genes with that extra target. And those genes are genes that come from public databases, including ICCG, but we also add up to about 2,000 genes from our own manually curated database. And those are genes that aren't yet considered to be medically relevant, but likely will be in the future. And so in that effect, we're really future-proofing our gene target for ACE. ACE isn't limited to exome sequencing. We're also targeting key intergenic and intronic regions that are also of medical relevance. And so we're going beyond standard exome sequencing in this design. What I want to show you here is a little bit more about how ACE actually works, or how this additional capture works. And so just to sort of orient you to all these different blocks that you see on the screen, if you look at the yellow blocks are really representing for any given gene exons. What you see in blue is what would be covered by your standard exome sequencing capture kit. And so the first thing that you note is that you may see some that are covered well, some exons that are more or less completely covered. You're going to see some that aren't well covered, and then you're going to see some, particularly these first exons, that aren't really covered at all in some genes. What we do with ACE is we actually target on top of these areas that are poorly covered. And so what you get with ACE is actually the blue plus the green. And so when you add those two together, what you end up seeing, if you're comparing an enhanced exome to these exons here, what you're seeing is much more complete or comprehensive coverage, to where we could say that we complete or finish the exons across any given gene. And as we also discussed just a moment ago, ACE also targets regions outside of the exons. And so that's what you see here represented by these red triangles, is that oftentimes if there's medical relevance in a certain intronic area or region in a gene, we're also providing additional targeting towards those regions as well. So back to talking about the methods from our paper, these are just some of the technical specifications listed here. The one I want to point out specifically for exomes and ACE is that we downsample data to 100x coverage, average coverage, and 12 gigabases of data across the whole target set for each of those exome platforms. The effort that we took in doing that was really to help normalize the data as much as possible across their complete target set, because it allowed us to compare platforms more equally, since there's so much variation in the complete footprint of each of these different assays. Just for your own reference, the slides that we'll show in the next few moments and the figures that we'll talk about, those are all based on the downsampling to 100x average coverage. We also, as I mentioned, downsampled to 12G and compared, and the data were really consistent, looking at 100x and 12G, and those data are also available in an additional file. So the study focus, as I said, is clinical in nature. We wanted to assess performance over clinically interpretable genomic regions, and we also wanted to look at performance characteristics that were required for making correct diagnoses, as well as minimizing the risk of misdiagnoses. And so we looked at a few different parameters here. We looked at something called gene finishing of the medically interpretable genome, and those are both concepts that I'll go over in the next couple of slides. In addition, we also assessed coverage of difficult to sequence but biomedically important regions of the genome. And last but not least, we specifically looked at coverage of the 56 genes that are recommended by the ACMG for reporting of secondary findings. So I want to spend a little bit of time to talk about gene finishing because I think it's a concept that unfortunately isn't commonly used in clinical diagnostics, although we use it at Personalis quite a bit. So just as a brief background, NextGen sequencing, typically what you're doing here is you're obtaining millions of strands of sequenced DNA that are around 100 to 125 base pairs in length. And those sequences are aligned to a reference genome. After this, what you see is that some bases are going to be very well covered, but you may have some areas that are poorly covered. Most labs really focus on obtaining average coverage across a given gene's coding bases. However, recent publications have shown that you really need to have at least 20x coverage in clinical diagnostics to have high confidence. And so when you say average coverage, you may have an area that has 4x coverage, and you may have an area in a gene that has 40x coverage. But you're still going to have a number of areas that fall below that 20x threshold that you really need in clinical sequencing. And so at Personalis, we use a concept called gene finishing as our metric for determining coverage, which allows us to really strive for getting complete gene coverage across a gene. And so we consider a gene to be finished when 100% of the coding or exonic bases are covered at 20x or greater. And so this might seem like a really high standard to shoot for, but we understand and we believe that it's really essential or critical in clinical diagnostics for really achieving accuracy. So to evaluate relative platform performance, we calculated coverage and accuracy statistics for ACE and other platforms within these commonly defined medically relevant target regions. And so accuracy was assessed by using two different gold standard data sets or call sets that are available for NA12878, again, that common reference that we're using. First, we used the high confidence call set. And this call set is looking at regions of the genome where the likelihood of an incorrect call is increased. Those are actually removed from this high confidence call set. And so this is really restricted more to high confidence regions among multiple platforms, aligners, and variant callers. And so given that, we also utilize the second call set. And that call set contains variants that fall into difficult to sequence regions. And that call set's really useful for evaluating regions that are typically excluded from these high confidence call sets that we've already talked about. And that might be an area, for example, of high GC content that may have been removed from a high confidence call set. So as I mentioned, we looked at gene finishing and call accuracy across what we call the medically interpretable genome, or MIG. This medically interpretable genome is a set of 5,419 genes that have mutations in genes implicated in human disease or disease-related drug response. So these genes are often part of an existing clinical test. They're documented in the literature as pharmacogenes, or they have a causal association with Mendelian disease, inherited disease, or cancer. As you can see from the figure at the right, the majority of these genes are coming from three main public sources. And so as you see here, HGMD, OMIM, and the Genetic Testing Registry. We also supplement the remainder of those from the Cancer Gene Census, as well as PharmGKB's list of VIP genes. And these are genes that have dosing guidelines associated with them. One point that I really want to note here, since we're talking about the medically interpretable genome, is that there is a difference in this medically interpretable genome and our list of the 8,000 genes that we're targeting with our clinical product that we offer. So I just want to point that out because for the purpose of this study, we really wanted to ensure that all of the genes that we were looking at as part of this MIG, that these were genes that were all part of existing clinical data sets or public data sets, so that there's no disagreement whether or not any of these genes are really medically important. So just keep in mind as we're talking about the medically interpretable genome for the purposes of this publication, it's a much smaller data set of around 5,400 genes as compared to what we're actually targeting with our clinical product, which is closer to 8,000. So now let's move on to the results and talk a little bit about what we actually found in the study, since now we've covered all the background leading up to this. And so I'll orient you to the different figures as we go on. But in this case, what we're looking at is coverage efficiency in the medically interpretable genome. And so for these stats, again, looking at those 5,419 genes, the left panel, what you see here, is looking at efficiency for the coding regions by platform. And on the right side, we're looking at the efficiency in the non-coding regions by platform. And so each platform has its own designated color, and the acronym is right beside it, so you can easily tell which one's which. So going over to the left graph, what you see here is the percentage of medically interpretable genes with greater than 90% to 100% coverage of exonic bases. And then on the y-axis, you see the coverage depth from 0 to 60x. And so what I want to draw your attention to is the dotted line that you see here that's representing 20x, which again is that clinical grade standard that we've talked about already. So when you look at the actual coverage looking at 20x, we see on this graph that ACE has the highest amount of coverage efficiency, followed later on, which looks to be a tie with whole genome sequencing and NextGen. And so ACE comes in around 99% coverage efficiency here, and whole genome sequencing and NextGen are approximately 97%, and so on and so forth, as you see by the rest of the platforms. On the right-hand side, we see a different picture, of course, and so we're looking again at coverage depth using a slightly different mark here from 40% to 100%, and again, depth from 0 to 60x again. And so again, when you go back and look at this 20x coverage, you see in this case, whole genome sequencing is coming out on top in terms of coverage efficiency in the non-coding regions. But very quickly, by whole genome sequencing is ACE. And then as you continue to go down, we see much lower coverage efficiency in the non-coding regions. This is SureSelect SureSelect version 5 with UTR, followed by the rest of the exome sequencing platforms. And this really isn't surprising when you think about it, since most whole exome sequencing platforms really aren't targeting this non-coding region. And so that would make sense that whole genome sequencing ACE, and then SureSelect, which has some coverage in the untranslated region or UTR, are going to be sort of your top three in terms of non-coding region coverage. So we also looked at that concept that we've mentioned a few times already called gene finishing, and again, looking at the medically interpretable genome. And so what this is looking at on the left-hand side is the relationship of the percent of finished exons, which is on the y-axis, as the coverage stringency varies on the x-axis. And so you go here from 90% to 100%, exonic bases covered at 20x or greater. The right graph is showing the percentage of finished exons with 100% base coverage as the read depth varies from 10 to 20x. And so essentially these are two ways of looking at the same data. And so either way you look at this data, what you see is that at these higher coverage stringencies, again 100x on this, or 100% on the left-hand side, you see that ACE finishes more genes than whole genome sequencing or whole exome sequencing assays. We're finishing with ACE approximately 90% of all of those 5,400 genes in the medically interpretable genome, compared with anywhere between 30 and 65% of genes finished with the rest of the whole exome sequencing platforms, and only about 10% of genes are actually finished with whole genome sequencing. So as I mentioned earlier, we also wanted to look at some of these very difficult to interpret genomic regions. And so with ACE, we're really targeting and capturing these difficult genomic regions, such as high GC content. And so we wanted to compare that performance across, again, whole exome sequencing platforms as well as whole genome sequencing. And so here, what you see on the y-axis is the percent of finished exons in the medically interpretable genome. And across the x-axis, what you're seeing is the actual percentage of GC content. And so what this is showing is that as the GC content increases, you start to see the amount of gene finishing in whole genome sequencing and other platforms really start to significantly drop off and be able to finish much, much less, right around when you get to about 50% GC content in a given region. However, when you look at the coverage by ACE, you see much more consistent ability to really finish these genes despite having those difficult to sequence regions or this high GC content. And so ACE really holds on much stronger in these difficult to sequence regions. So this slide here is a little bit busy. So what I want to do with this one is kind of break it down in a few sections. And I've got a few extra slides to sort of help illustrate some of the main points that we want to talk about with this particular slide. So as you probably presumed, this medically interpretable genome that we've talked about, it includes, of course, the 56 genes that per ACMG guidelines are recommended for reporting of secondary findings during clinical genomic testing. So of course, whole exome sequencing and whole genome sequencing would fall into this category. There's a number of papers out there that have really highlighted the inaccuracies in NextGen sequencing with regard to detecting variants in these particular genes. Since these genes are well known in the medical setting, they've been well studied with regard to NextGen sequencing. So these inaccuracies that are sort of well known out there can be really misleading in the clinical setting, because the absence of a variant on a clinical report in one of these genes might provide false reassurance to a patient, a clinician, a family. The problem is that the actual coverage in these genes with any given platform really varies. And so a negative result doesn't, of course, mean that the gene has been consistently studied, even with whole exome sequencing or other technologies. So this is really the exact concept that we wanted to look at in this case. And we saw quite a wide range of coverage in these genes. Exome-based platforms generally performed better than whole genome sequencing as a whole for these 56 genes. However, none of the platforms that we looked at, including ACE, our own platform, were able to fully cover or fully finish all 56 of these genes. And so there was certainly inadequacies in all of these areas. And so if you look down here at the bottom, this is really looking at the stats for gene finishing for each of these platforms. And so we did see that even though none of them covered completely, we did see that ACE was able to completely finish the most of these genes, and so 51 out of 56. Whereas if you look at the rest of the other 4 whole exome sequencing platforms, you see coverage anywhere between 12 to 39 of those genes finished. And with whole genome sequencing, only 2 of the 56 genes were completely finished, looking at the ACMG genes. So now I want to talk a little bit about this figure at the top here. And so in this particular figure, we're actually not talking about the entire coding region of each gene, but rather we wanted to focus on known disease-associated variants. So what I did was I took a snapshot of this and sort of zoomed in a little bit, just so it can be a little bit more clear on the screen and we can really talk about what's going on here. So we talked about disease-associated variants, or looked at that for the ACMG genes, because we noticed that several of the platforms that had inadequate gene coverage had inferior coverage in regions where there were known gene variants. We actually looked at around 12,000 documented variants in these genes, and we determined the sequencing coverage with all of the various platforms. So for whole exome sequencing, for each gene, we actually only plotted the platform with the highest amounts of coverage, just trying to keep the slide a little bit less confusing. And so what you see here, in terms of the key, is that the square that you see here is representing whole genome sequencing. The green triangle is representing ACE. And the circle that you see here, again, is representing the exome sequencing platform, the one with the highest amount of gene finishing. And of course, that may be different for any given gene. And so there's much more full information in the supplemental material of this article that breaks it down by each specific platform. So what I want to point out here is that every platform, as you can see, there were coverage pitfalls in looking at variants in these genes. And so, you're seeing, a lot of coverage pitfalls with whole genome sequencing, but you're also seeing some triangles and some circles that fall below this 100% variant finishing area here. And so what that amounts to statistically is that overall, those coverage inefficiencies were as high as about 9.6% with one of the whole exome sequencing platforms. It was around 6% with whole genome sequencing. And for ACE, only about 0.2% was what we were looking at in terms of coverage inefficiency of these particular disease-associated variants. What I want to point out, in case you haven't seen it yet, though, is one specific gene that looks quite troubling across all of the different platforms, and that's MSH6. And so again, with MSH6, you see very low coverage in terms of whole genome sequencing. 95% of the disease-associated variants were completely finished with whole genome sequencing. and around 98% with ACE and one of the whole exome sequencing platforms. And so what I want to do now is sort of go into a little bit more detail about MSH6 specifically. And so what you see here is something that we use at Personalis quite a bit. So anyone who's familiar with Personalis, I'm sure has seen these blue-green plots quite a bit. What this is a coverage plot. And so it's a diagram that plots out gene coverage across either the gene's coding bases or the gene's full content. It just depends on what plot you're looking at. And so what you see on the y-axis here is depth of coverage, or again, that's X. And so this goes from 0 to 20X to 40X to 60X and on up. The red dotted line we put in place so you can really keep an eye on this 20X, or this gold standard level of coverage that we really strive for in the clinical setting. And so what you see here is the blue coverage is really what's provided by a standard exome sequencing capture kit. So any of the 4 that we've talked about, you could sort of use as an example of what this might look like for any given gene, with some variation, of course, in coverage. The green area is what we add to the picture. And so the green is sort of Personalis' augmentation or additional coverage. And so while a standard exome would be the blue area, a Personalis exome would really be the blue plus the green. So now that you kind of have an idea of what's going on here, I can tell you what we're looking at. And so what we're looking at here are 2 different gene plots for MSH6. The top is the coding areas only, or coding regions only. These are the exons that are sort of concatenated or smushed together on this plot. And when you look at the coverage here, you actually see really great coverage at 20X, all the way across the coding regions. And so this is a great gene that really sort of shows that there's not much that we need to do at Personalis to augment this gene from a coding region standpoint. This gene is really well finished on its own with many of the standard exome sequencing kits. However, when you go down and look at the full content, that's when you see a little bit of a difference. This is the full content of the gene, and it has the exons included, of course, but it also shows the other areas outside of the exon where we would be targeting specifically. And in this case, you see 2 areas where we're specifically targeting or adding that green. And this is because we know that there's variants in these particular regions that are outside of the exon. Unfortunately, what I want to point out here, which goes back to that figure that we showed in the prior slide, is that even though we're targeting in this additional region here, we're still failing to reach that 20X level of coverage, even with our own supplementation. And so that really speaks to this being an area that's really difficult for all platforms to sequence, including ACE. And so this really explains why that particular gene might have low finishing stats for these disease-associated variants across every single platform, because if we're targeting to get additional coverage and still are unable to fully finish this particular area, then certainly the other platforms would be having the same issues. So to summarize the article, most platform comparisons have really focused on entire exomes or genomes, and this is the first study that's really looking solely at genes of medical relevance, which is an area that's specifically applicable to clinical care. So as we've discussed in previous slides, the ACE exome offers a number of advantages in this area when compared to standard whole exome sequencing platforms, as well as whole genome sequencing. This is important for both Mendelian applications, but it also has utility in cancer research and tumor profiling, and these are applications that are also supported by our ACE technology. So what I want to do next is go into talking about how all of this really plays out in the clinical setting. So, one of the things as working as a field application scientist at Personalis is, we're oftentimes meeting with geneticists and genetic counselors in the field and talking about our technology. And one of the common and completely appropriate questions that people ask us at the end of a presentation is really, it's great that you're providing this extra coverage, but does it really matter? Does what you're targeting or what you're augmenting really make a difference in the clinical setting? And so what I want to show you here is really a couple of different ways that our ACE enhancements play out in the clinical world. And so one side, on the left-hand side, this is really showing that, for one, approximately a third of the variants that we report clinically are variants that are outside of what's commonly assessed with a conventional exome. So these are variants that we might specifically target for enhanced coverage that might not be well covered at all by a standard exome, or they may be areas that aren't covered by a conventional exome at all. So of course, this is one area where our ACE enhancements are really helpful. Another sort of way to look at it is by looking at it in terms of the diagnostic yield. And so we've seen a higher diagnostic yield, approximately 47%, when compared to some of the other clinical labs. And this is in large part due to the improvements on the sequencing side that we've talked about. But one other part I should point out that we're not going over for the purposes of this presentation is that at Personalis, we've also improved the process from start to finish. And so that entire ACE platform are areas where we've innovated in alignment, variant calling and annotation, as well as our interpretation services. So all of these sort of pieces have led to this increase in diagnostic yield, not just the sequencing as we're talking about today. So we think of our ACE clinical exome as really a single solution compared to multiple separate tests. There's the broad coverage that you'd get through exome sequencing, of course. But as we've talked about quite a bit, we also focusing on finishing these genes, similarly to what you might see in single gene or panel testing. And though we haven't talked about it today, and it wasn't part of this discussion, we should also note that our ACE technology, we have proprietary methodology that also includes detection of large structural variation. And so ultimately, although the actual cost of our ACE exome sequencing really falls in line with what we see on the competitor landscape, we actually end up providing much more than a standard exome here, really an exome plus, as you might think. So again, what does all this mean in the real world, right? There's a couple of case examples that I'd like to kind of go over to sort of illustrate all of these points that we've really talked about up to this point. The first example I'll show you is a pair of identical twin brothers who had been diagnosed with infantile onset retinitis pigmentosa, high myopia, and there was no additional family history. These were the first individuals in the family to have been diagnosed with RP. Just like most patients who have exome sequencing, these kids had had some testing done before coming to their clinic for exome sequencing. And so in this case, one of the individuals had had a 13-gene autosomal recessive retinitis pigmentosa panel, and that autosomal recessive component will be important in just a moment. And they'd also had standard whole exome sequencing performed, and so that's not common. Of course, most people will do one whole exome sequencing test and leave it at that, but this was one where they'd had an exome that was negative, and the clinician was convinced that there was something that was being missed here. So as most of you probably know, retinitis pigmentosa is a condition that's extremely genetically heterogeneous. There's dozens of genes that cause retinitis pigmentosa, and it's inherited in a number of different ways, dominantly, recessively, as well as X-linked. Variants in a gene called RPGR cause about 80% of X-linked retinitis pigmentosa, which accounts for about 20% of retinitis pigmentosa as a whole. A standard exome, again, going back to these blue-green plots that we talk about, a standard exome, looking at just the blue here, would cover many areas at least 20x. But of course, as you see here, there's one specific area, this big hole in this section of the gene, where there's very poor and barely any coverage given by a standard exome. This particular area is an exon called ORF15, and it happens to be a mutational hotspot in this gene. So there are quite a few pathogenic mutations that show up in ORF15 of RPGR. The downside of that is that this is an area that's really notorious for being difficult to sequence due to the amount of GC-rich regions in this particular exon. And of course, that's an issue because in RPGR, about half of the mutations fall in this one exon. And so, as you can see here from a standard exome sequencing standpoint, this can be really problematic, particularly if your variant is in that half of the gene or the exon. And so when we look at the actual supplement with ACE, this is really what happens here. So again, we're looking at ACE with the blue plus the green. And so what you see at this point is that when we add our additional supplementation, what you end up seeing is really consistent coverage again across this gene. And so this, from our perspective at this point, would be a gene that's 100% finished across the coding regions. And in this specific scenario, it was really important. And so in this family, we actually did find the diagnosis. We found a mutation in this ORF15 exon that, as we discussed, was missed by the prior exome that was completed. This is important for two reasons. One, as I mentioned before, the clinician was initially going down the road of autosomal recessive inheritance, and we ended up finding an X-linked mutation, and so it sort of changed a little bit about what the clinician was thinking from an inheritance standpoint. More importantly, from an exome sequencing standpoint, it was in an area that was missed by a standard exome. The second case I want to talk about is one, excuse me, of a female who had a clinical diagnosis of Charcot-Marie-Tooth disease. Her father was more severely affected than she, and in this particular case, we were unaware of any testing that had been completed prior to the patient coming for exome sequencing. What we identified in this case was a pathogenic variant in a gene called GJB1. This variant and gene are associated with Charcot-Marie-Tooth disease of the X-linked type. So, of course, her findings really, of course, fit her phenotype, it fit her family history, so everything really made sense from the standpoint of the variant that we detected in the gene that we found in this family. But what was really interesting from a sequencing standpoint is where the mutation was found in this gene. And so what you see here, again, another one of these famous blue-green plots, what you see is that this gene is right here in the front of this gene in the promoter region. What's important about that is that standard exomes, of course, are not targeting the promoter region in their design. And in fact, a number of next-generation sequencing panels for Charcot-Marie-Tooth disease or hereditary neuropathies, they also don't target the non-coding regions. Certainly some of them do, but many of them do not. So this became really important in this particular situation, of course, because the variant that we found was in this region that many other platforms would have not tested for. So even if this individual, again, we don't know the testing that was performed, but even if she had had exome sequencing, for example, performed in the past, it very likely would have been negative since this area would not have been targeted. And so, in summary, from looking at these case studies, what we really show here is that this enhanced methodology through ACE is really important for targeting some of these difficult areas. Again, we focus on what's provided by a standard exome, but also by targeting this additional coverage, it really makes a difference in clinical diagnostics, as you can see from these two case examples. So really summarizing today, there's a number of important conclusions that are apparent from our own experience at Personalis through these case studies, but also from the recent findings in the Genome Medicine publication. First, clinicians really should focus on analytical performance when making clinical assessments or choosing labs to perform testing. Even a few missed variants or a couple of areas of poor or inconsistent coverage can lead to reporting false negatives and as a result giving false reassurance. Personalis's augmented approach is an innovative strategy that provides coverage that's not available with other platforms. And so this strategy it's performed as part of our analysis. There's no need to fill in or say perform Sanger sequencing on sections of genes that don't have high coverage. It's not an extra process that we perform later on. It is our process. And in the clinical setting, this augmented approach to exome sequencing, specifically focusing on these areas of medical relevance, it can be cost-effective, and as a result can lead to diagnoses earlier and with fewer tests than your current or standard genetic workups. And so now that concludes the presentation portion.
Thank you. We have now come to the end of our time today. Thank you all very much for joining us. We hope that you all found this presentation beneficial, and again, extend our thanks to Heather for leading this informative and interesting session.
Thanks again, Felicia.